Okay, so this is, I know these are wrong. Okay, so this is incorrect. This is from a different book. So this is chapter 7 and 8. So 7 and 8, I'm going to combine together in one PowerPoint. So first thing is, this is all about microbial genetics. Okay, so it's the genetics of bacteria. Okay, so the first thing is, what are chromosomes? Okay, so if you remember from your biology class, right? Chromosomes they're kind of like packets of genetic information. Okay, so they're made out of DNA, right? And there's proteins wrapped around the DNA, so that's how it gets all combined, and that's how it forms chromatin. So chromatin is kind of when it's loose and not condensed. So now prokaryotic chromosomes, okay, so bacteria, most of them only have one single circular chromosome. Okay, so one. So if you think about us, okay, we have 23 pairs, right? So we have 23 times 2, that means we have 46 chromosomes. Bacteria typically have one. Okay, so big, big, big difference. Ours are linear, right? So if you think about ours, the X's, right? Ours are linear, but bacteria are circular. Okay, so it's kind of like this. Okay, but it's bigger than a plasmid, which we're going to talk about plasmids here in a minute. So now here is a picture of bacteria that's going through, a bacterium that's going through binary fission. Okay, so it's dividing here. This red stuff, so this is transmission electron micrograph, so that means it's been super duper magnified. Okay, and they colorized it, so they never look, very rarely, they don't look colored, right? So they go in and somebody colors it. So this red, right, that's the DNA in bacteria. Okay, now remember, in bacteria, there's no nucleus, but there is this region, it's called a nucleoid. Okay, so the nucleoid is this kind of condensed area where you're going to find the DNA, but it does not have a plasma membrane. Okay, now I'm going to, I guess, put this, let me put in a plain page. Okay, so I just want to put in a plain page. I don't remember how to do it. I guess I'm not in the right. There, never mind. I just didn't click the right button. Okay, so I have written down haploid. Okay, so if something is haploid, it means it only has like a single chromosome. So it's the chromosomes aren't doubled. So like we are diploid. So diploid means we have 2n. So we have 23 pairs, right? So we have 2 times this number. So we're diploid. So if you think about our eggs and our sperm, right? Those are actually haploid. So they have 23 and 23. Okay, so that's haploid. So bacteria are haploid. They don't have a pair. They just have one. Okay, where if you have cells with pairs, they're diploid. It's kind of crazy because there are some organisms that are like triploid or polyploid, so they have even more than 2. Let's go back here. Okay, now we're going to talk about plasmids. So sometimes bacteria have plasmids. I mentioned it before. We'll keep talking about it. Okay, now some vocabulary, which most of you are probably familiar with, but let's just make sure you are. So a gene is a piece of DNA or a segment of DNA, and usually it codes for one protein. So it codes for a protein. So it's going to be a piece or a sequence of DNA. So some A's, T's, C's, G's, right? And this code of DNA is a packet of information, okay? And it's going to tell your cell what protein to make. Okay, so a gene pretty much controls or codes for a protein. Sometimes they control protein. Okay, now locus looks like location, right? So it's location. So a locus is a place on a chromosome where you typically find a gene. So it's gonna, if you have like the gene for, well, everybody say, let's look at something. So everybody's got the gene for eye color. Okay, so the eye color gene should always be in the same locus or the same location on the same chromosome in every person. Okay, so locus is just where you find the gene. And you can look at a map. So you can look at a karyotype, which I didn't put a picture in here. So you can look at a karyotype. So karyotype is just a picture of chromosomes. So a picture of your chromosomes. So I can look here. Let's see if we can. I go here and I go to Safari. And then let me go look, find a picture for karyotype. Right, and we'll do this. Let's look at this. If I can make it bigger. Visit. Okay, so here is maybe it's gonna load. Here's a karyotype. Okay, so if you look at this, so this is a human karyotype, which I can't write on it. But so 1, 2, 3, 4, 5, 6. So 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22. Right? So 22 pairs of autosomes, which that's the information that we need to function. And then the last pair, the 23rd pair, those are the sex chromosomes. Okay, so if you look at this one, this is an X and a Y, so this would be a male. But a karyotype is just a picture, a representation of the chromosomes, and it can be a picture of them, right? And then the banding is showing like kind of where the genes are. So like if you have this band here, right? So you say you have this, where the banding patterns are, those are locations, those are loci. So if you look at this, see this red, well, that's probably the centromere. So maybe you could do the top line. So the top line should be in the same spot on both chromosomes. And if you looked at somebody else's DNA, they should have it in the same place. Okay, so now let's go back here. Okay, now an allele is when we have two copies of something. So an allele is like, if this is a gene, okay, so we have one allele here and we maybe have another allele there. So we could have different formats of the gene. Okay, so gene, locus, allele. Okay, now you should have learned this in general biology or in A&P, but I'm not 100% sure that you have. Okay, so I want you to know three big concepts here. So like, how do we get from DNA to a protein? Okay, so this is key. This is how our body functions. If this doesn't work, we have sickness, we have death. Okay, if it doesn't work, we're in bad shape. Okay, so three main processes that we're going to have. So the first one is replication. And if you look down here, you'll see replication is just when we copy DNA. So we're going from DNA to DNA. Okay, so we're copying it. So we're just making an exact copy of it. Okay, now this is going to happen in the nucleus. Okay, because we don't let it well, it's going to happen in a nucleus in a eukaryotic cell. It's just going to happen in the cytoplasm of prokaryotic cell, okay, because they don't have a nucleus. Okay, so it's happening just in the cell if it's bacteria. Okay, now transcription is when we take the DNA and we copy it or we transcribe it and we make RNA. Okay, so this happens either in the nucleus, right, or in the cytoplasm. Okay, if it's a prokaryote. Okay, so remember DNA has A's, T's, C's, and G's, where RNA has U instead of T. Okay, RNA has a different sugar. Okay, so RNA is single-stranded. Okay, so RNA is different than DNA. Okay, now the RNA, if you're in a eukaryote, that can leave the nucleus. Okay, and it's going to travel to the ribosome. Okay, so translation occurs at the ribosome, and translation is the process of taking the RNA and reading it and making it a sequence of amino acids, which are a protein. Okay, so replication, DNA to DNA. Right, let me find this. Replication, DNA to DNA. Okay, transcription, DNA to RNA, right? And then I don't have another color. I thought I would have another color in this. Okay, and then translation is RNA to protein. Okay, now here's a trick. So transcription and translation look the same, right? The words are very similar. It's easy to get them confused. C comes before L. Okay, so this is going to occur before, you have to have transcription before you can have translation. So that's just a trick. Okay, now this is called the central dogma. So in biology, we don't have a lot of rules or laws. Okay, but this is one that we say, okay, you go from DNA to RNA to protein. That's the central dogma. So what I have in yellow down here. Okay, now it turns out, being biology people, right away they find, well, not right away, but they find that, okay, we're saying we have this law, but it's already been disproven. Okay, so there are some viruses that can do this process called reverse transcription. So reverse transcription means that they can go from RNA to DNA. Okay, so they're doing it backwards. Okay, and the example, HIV. Okay, so HIV has an enzyme called reverse transcriptase, and it is an RNA virus. Okay, so what it does is it takes RNA and makes it into DNA. It does the opposite, okay, of what we traditionally thought happened. Okay, now I just put one slide in here, and this is replication. So in prokaryotes, replication, remember, it's just going to occur in the cell, right? It's occurring in this basically in the cytoplasm. Okay, here is a nucleotide. Okay, so here's the sugar, here's a phosphate group, and then the nitrogenous base. So this is what an A, T, C, or G basically look like. Okay, and the only part that's different on it is going to be the nitrogenous base. Okay, so that's going to determine whether you have A, T, C, or G. Okay, so all DNA is, right, is a series of these linked together. okay, and it's double-stranded. Okay, so if it's double-stranded, that means you have a complementary base pair on the other side. So A always pairs to T and C always pairs to G. So A to T, I just remember a trick I tell everybody is straight edges go with straight edges and curved letters go with curved letters. Okay, so C is going to go with G and so on. Okay, now the way that replication occurs is basically it's a series of enzymes, and you have A's, T's, C's, and G's in there, and the enzymes are going to break apart this stranded piece of DNA, and the complementary base pair is going to come in here and bind, and we'll have another enzyme that ligates it together. Okay, when we make DNA, we go from 5' to 3'. Okay, so we're going this way. Okay, so if we have complementary DNA, so this is 5 to 3, and this one is going to be the opposite, 3 to 5, right? So what happens is one is going to get read in a continuous strand, and the other one's going to get read in these little pieces. And those little pieces are called Okazaki fragments, and those actually get pasted together. They call this one that's complete the leading strand, and the one that's broken apart, they call it the lagging strand. Okay, so you end up with a copy. So you end up with 2 pieces of DNA from the original sequence. So this is showing you the original. So you have a blue and a purple. Wait a minute, let me try to highlight that. You have a blue and a purple. That's the original strand, right? And then the new DNA it is this orange. Okay, so you're going to copy both halves. So then you're going to end up with 2 sets, right? So you end up with, wrong color. Well, you could do it like this, a set that looks like this, so a new and an old, and then you're going to end up with, let's say, the purple, so a new, or an old and a new. Okay, so that is called semi-conservative replication, because you're conserving the old, but you're making a new from it. Okay, so it's half and half. Okay, now the 5' and 3' thing. So the 5' and 3', that's based on the carbons in the DNA, or not in the nucleotides. So when you do biochemistry or organic chemistry, they number the carbons. So that's how you know which one is which. Okay, so you just number them. So on this picture, so here's 1, 2, 3, 4, 5. Okay, so that's the 5th one. There's the 3rd one. Okay, so it's how they're oriented in the DNA strand. Okay, now, here's just another picture. So this is a TEM. Oh, so this is like real DNA in a cell. And they're trying to get you to see, which this isn't very clear, but they're trying to get you to see that, okay, you have some original DNA, and then you have copies of it. Okay, so you're going to end up with this original piece, and then you'll have two copies of it. And that's what I tried to draw on that last picture. So you have an old, right, and a new, and an old, old and a new. Okay, now the big difference right here in replication, right, is that this is circular. So when we have replication in eukaryotes, right, they're linear. So you're just copying, but it's going to be a ring, a circle. Okay, now I made a little chart because that's the way I roll. I like charts, right? So, and this just summarizes it. So the location of the chromosome. So in prokaryotes, it's in this spot called a nucleoid. right? So there's no membrane-bound nucleus. Okay, and the eukaryote sits in the nucleus. Okay, most of the time in prokaryotes, they're going to be circular, and there's only one, and it's haploid. So they only have one copy of a gene. Whereas eukaryotes typically have two. Okay, they have different numbers of chromosomes depending on what the organism is. So some have more than we have, and some have less. And they're usually linear. Okay, now, transcription and translation. So in a prokaryote, okay, so in bacteria, transcription and translation can occur at the same time. So while you're copying DNA, okay, and you're making RNA, it can travel to the ribosome, which this is a very good picture, and then we can make protein. So all of this can be occurring simultaneously, right? So there's no nucleus. The DNA doesn't have to leave the nucleus in order to get to the ribosome. So everything can be happening at one time in the cell, which is really cool. Okay, now they're also called polycistronic. Okay, so polycistronic means that genes are read and can be read in like little groups. Okay, so poly means more than one. So more than one gene can be under the control of this thing called a promoter. Okay, so a promoter is what turns a gene on and off. In a eukaryote, so let me make it blue. In a eukaryote, there is one promoter to one gene, and one promoter to one gene. Okay, in bacteria, there's one promoter, okay, but it can control many genes. It can control more than one at a time. So this is going to be what tells the gene when to turn on. Okay, so when we need to make it. Okay, and lots of times these genes are related. Okay, so they're all going to work basically in a pathway together. So we need all three of them to do the job. Okay, so if we turn all three of them on, it kind of saves energy, right? So you say, oh, I need to break down lactose. Okay, so I can turn on this sequence of three things that are going to break down lactose, and I don't need just one, I need all three. So it's actually kind of slick. It's pretty cool. Okay, now, how do bacteria control when genes are turned on or off? Okay, so in anatomy, you learn about expression, and in Bio 113, you learn about like what turns genes on and off. Okay, so there are different signals that might be in the environment or are coming from another cell. Okay, so if you think about like a neuron, and a neuron releases a neurotransmitter, and that turns the cell on, and then it starts producing the next thing that we need, or a hormone might do that. Okay, so it can be a signal from the environment, a signal from another cell. Bacteria have this thing called quorum sensing. So quorum sensing is the idea that when we get a certain number of organisms, so when we reach this set point, they will produce enough of a chemical or enough of a signal where they will turn on gene expression. Okay, so a quorum is basically a number. So like if Congress votes, for the vote to be legal, they have to have a quorum. They have to have a certain number of members there. So if nobody's there, they can't pass something that everybody doesn't agree with, right? So bacteria do this thing, they call it quorum sensing. So you're waiting and trying to accumulate a certain number of cells in order to turn on a gene. So it's interesting because they've figured out that with some diseases, you need a certain number of cells to accumulate, and the cells have to be able to communicate with each other, right? So talk to each other, and then they might produce a toxin that will cause a disease. So what scientists are thinking, right, is that let me try to get a different color. If we can block these and stop them from actually communicating, it won't matter how many are there because they won't ever produce this toxin and they won't cause disease. So they're trying to regulate and control quorum sensing to stop the bacteria from causing disease. So it doesn't matter how many you have in there if they don't make the toxin. So it's interesting. Okay, now a constitutive enzyme, that is an enzyme that's always on. Okay, so it's stuff that you're going to need to survive. So constitutive just means it's always turned on. Okay, inducible means we can induce it, we can turn it on. And then repressible means we could add something to it and turn it off.